bit to get used to. I might forget. I'm George Malone. I received the supervised. This evening I'm talking just in about 3 or 4 weeks. So, first, a little bit of background. Based compounds. Here we focus on C and to form groups known as CPG islands, and CPG islands in terms of DNA methylation. And finally, there was the methylation in gene promoter region are correlated with diseases like cancer, well, or Alzheimer's. So, one branch which converts unmethylated lysozymes to uracil. This is PCR amplified, but it's uracil main methods. Array, treated and untreated sample are added these probes, which when they react with DNA, they fluoresce, producing red light for cytosine, green light thymine. Your resulting measurement is the ratio of red to green fluorescent intensity. Another method, the sequence pre and post conversion, or rather the DNA pre and post conversion are sequenced, and at each location in the genome, the count of reads of cytosines and thymines are counted. Cytosines pre and post. The resulting measurement is the ratio of post to pre cytosines because cytosines are methylated cytosines are not converted. That also tells you the degree of methylation. The results data for every site in the genome and every, shown by the rows here, and every sample, get a numeric value between 0 and 1. So, methylation ratio is a model for estimating the biological age of an organism called the epigenetic clock. Fundamentally, these are linear regression models, which are generally trained on array data via the elastic net. These have applications in agriculture, pressing on the difference between estimated and true age, a value called age acceleration. Applications in health medicine. More recent models are able to perform event prediction for development of cancer or mortality. So the elastic some subject to a constraint on the coefficient is controlled by 2 parameters, alpha and lambda. Alpha is the and lambda is the scaling weighting parameter. Special cases are when alpha is equal to 0, the ridge regression, and alpha equals 1. So shown here are the ridge on the right, massive constraint area dimensions. Ridge coefficients are shrunk towards, but not exactly 0, curved surface. When alpha when alpha is greater than 0, those sharp points mean that you can form feature selection. The coefficient values are set to exactly 0. Feature selection is especially useful in cases where you have the number of parameters greatly exceeds the number of samples, such as if you are measuring DNA methylation data. So most epigenetic blocks are trained on array data using a particular method, but sequencing is becoming more affordable and becoming more popular because these sites that are outside the range of 6 sites. So, can sequencing data be substituted for array data in array-based plots? A primary question. For this one, of paired data, we have paired Illumina 450K array and whole genome by sulfite sequencing 23 German pediatric brain cancer samples. Available International Cancer Genome Consortium, and they represented all of the samples that they actually had with paired arrays seeing data. So, the collection of 6 epigenetic clocks, we compute the age estimate for array, and for sequencing, we're looking for linearity, looking for any systematic or extreme difference. Quick look at the data, the distributions of data in particular. You can see that they have a common range between 0 and 1. They both sort of concentrate towards the extremes. Sequencing concentrates at the extremes at 0 and 1, whereas array tends to hover around 0, 1, and 0.1. Range estimates for the 6 clocks that we'll be looking at. Array is on the Y, sequence is on the X, and the solid blue line Y equals X is our target. The dotted line, linear model for illustration. Linearity is overall not too bad. And the error rates, remember the RMS in the lower right corner, also not too bad. Maybe. Shivey and Jang are near perfect linearity, but they're offset by constant amount. Good, but can they be improved? Transformation. Gene expression is a related field in epigenomics, which has seen a similar trend towards sequencing data. And the sequencing data also count data like sequencing. Function boom, our package lemma, uses a transformation called log2 count. Gene expression. You can see straight away that it hasn't worked. Right? Two-dimensional density of the transform data. Again, the solid line is what we're aiming for, and it's very far off, and the variability increases as you increase over the range of array data. So we don't chase this up anymore. So linearity is quite reasonable, but this is hard to judge with only 23 samples. And methylation is also regulated in cancer tissue. But the question is it more variable as well? And so those could be affecting the results, but this is a relatively limited concern because we're trying to do a direct comparison between platforms, not comparing to true age in this. So even if the methylation is dysregulated, the value should theoretically or hopefully you can transformation may be invalidated by the change, or it simply doesn't apply in this context. So further work here, we can do more transformation. Are there other existing transformations? Is there a novel transformation that we can learn? Would we apply this to array data, or will we apply it to age estimate? For example, Shivey and Jang, they were linear but off by something. Post-computation. Otherwise, simulation to increase the biased to a small sample size, and it would also be biased to the fact rather than something like blood in a healthy patient is probably what. So even and there are multiple different array platforms. Three, I think, coming out quite soon. Coverage over the same CPGs may not be guaranteed between these platforms. So how sensitive are epigenetic clocks to missing data? We'd expect the errors to increase as the data loss increases. If you see anything else, it'll be strange. So for this one, we don't need paired sequencing data. We can use a larger set of array data, the Brisbane study, collected method by the Illumina 450K array on blood, 614 individuals, twins, siblings, and parents. Same clock as previous, but with one CPG removed from the Zhang clock, later, or discuss why. The sample is missing the required CPG for each block per block basis, but each block has a different sample. So each block, we compute the full model age estimate, randomly remove some proportion. Compute the reduced model estimate, do this 1,000 times. To explore it, we'll take a look at densities of age estimates, errors per age. So here are the densities of the age estimates. As the data loss increases, the densities in the structure spread out and move towards lower value, except for Levine and Zhang, which move upwards probably because they tend to be quite close to zero or even, as you can see. Bimodality here is also an artifact of the fact of having parents and children, or I guess parents and offspring, I guess. I'm not sure whether they're children. Some of them are quite old. And so it lends the bimodality. Bang, in particular, I'll highlight again that it could have an also high analog and keep normal, fixed normal shape. The errors, median and variance increase universally, which is not unexpected. Most clocks keep a certain right unit, concentration towards lower errors, but Zhang degrades entirely. Levine as well shows an extremely high error. It goes all the way up to 100. Out of here, just the other clocks. See a bit of a similar picture for the errors against the true age, but here we see something particularly odd. A point. And this is heralded by the mostly negative values for Zhang for the full model and for very little data loss. All others, again, they completely dissipate. And again, Levine reaches all the way up to 100. So the result of it, increasing data loss increases error. For strange results for Zhang, models lost that skewness that was seen in the other plots, and the age estimates shifted closer to the true age as the data loss increased. So the removing the CPG from Zhang, fumble over my cards, it's the main concern here because 80% of valid samples for Zhang were missing this one CPG. And so in an effort to maximize the sample size, I removed that CPG, but adding it back in and dropping down to only 66 samples, only real difference, any major difference, is that maximum age is much larger for this. Otherwise, not much has changed. So not much is gained by adding a lot of samples, but not much lost by removing this one. The value, I think, was negative 0.015, so it doesn't really contribute much. So further work here would look at more clocks, such as those for age acceleration or the risk estimators that I mentioned before. But in particular, we can do imputation. Very similar procedure to this one here, except impute the CPGs removed instead of looking at the behavior of the clock. So Delana and others, 2019, did something very similar to this, a very similar routine, but with less proportion of error loss and only one clock, only the Horvath 2013. So we could validate their results in Brisbane sample. We could use a meta sample, like what they did. I'm not sure if theirs included Brisbane systems, but it may have done. And multiple clocks, especially more modern clocks, compared to the originator model. And that is all. Um, negative values, negative coefficients. So as you start dropping them out, the values start to be dropping out a lot of the negative values. That's a possibility. The better. Yes. And funnily enough, when you're actually using that particular clock, there is another version of it, the best linear unbiased predictor, which I haven't heard about yet. This one's the elastic net version. There are technically two versions. The best linear unbiased predictor is, well, it's actually about 1300. But it even comes up a little comment saying that if you have a lot of data loss, that it's best linear unbiased. So it's interesting that it even pulls you up on it without dropping data out. I guess in particular regards, but in the case of generally an elastic net, I guess it's because you're limiting your scope from 480,000 plus sites in the genome down to about 300 to 350 that are selected by, for example, Horvath's model, 360 something at an alpha value of 0.5, but not even fully elastic regression. And that was based on 27K and 450K, but restricted to the 27K. So you still got 26,500 sites. So I guess it's a way of allowing you to target CPGs. For example, if sequencing clocks really are quite linear, or sequencing data in clocks allows it to be quite linear, you could even do reduced or targeted sequencing. You wouldn't have to assay an entire region of the genome. But sequencing is quite expensive. Horvath worked on making a 40K chip, which was for targeting sites represented across pan mammalian, across multiple different mammals, not just humans. So that's a way in which it could be used as well, is restricting to different arrays, different kits. So yeah, my name is Alistair. I'm going to be talking about optimal contamination. Um, a rough outline for my talk is the problem and what the problem is that we're trying to solve. Talk about optimal estimator, about smooth and asymptotic analysis, which is of our work. And if we have time, talk about how this applies to data, summarize everything. Okay, my button is not working, so I'll click straight on. Yeah, so really quickly, a bit about me. I'm doing a research master's with training at Murdoch. Brenton is my main supervisor, Brenton Clark, and hat tip to him. He's been an inspiration, a mentor throughout the process. I also work full-time as a software engineer. Previously, I did an undergrad degree at Murdoch as well, which I did full-time while working time, which made me very busy and I'm told slightly mad. So anyway. I've got which is recorded as deviations in nanoseconds from 20 nanoseconds. And if we take this data and we make a histogram, And what you see on the right there, it's a normal distribution. And so we might say, okay, let's see if we can fit this and do some parameter estimation here. We can then estimate sigma height, which is location and scale respectively. And then that gives us an structure, just what these estimates are, but we understand a bit more about the data and we can apply probability theory in various ways. Of course, we've got sample mean, sample variance and standard deviation, or equivalently as maximum likelihood. And so we do that and we get our estimate to present it up there. And what we hope to see is when we plot this as a distribution, something like the green curve here, and what we actually get is more like the red curve. Wrong. And I'm supposing that most of you know what went wrong. We have some probable outliers on the left. And when we use parametric statistics, we make strict assumptions about the distribution. And in practice, quite often assumptions are not actually met. And it comes through various sources. Again, I'm going to skip through some of this because I'm assuming you have some background with this. But really, it can be a single errant observation that invalidates our model. So what can we do about it? Three options here that we're presenting. First, we could use the absolute deviation. We could manually remove the outliers. Robust estimation procedure. So why not just use the median as a starting point? Median is in fact the most biased robust. However, it has lower efficiency and is not as optimal on various robustness measures, which we'll present in a second. So we can do better, but to give you is from 0 to 10% on this. Okay, why don't we just remove the data that we've presented? That would requires domain expertise to actually understand what. And it may just be that we have a mixture of distributions. Normal denoted by capital phi. We have contamination. And let's just suppose, for example, the training process is also a normal distribution, which is often the case. And we can parameterize this. John's shaking his head. I realize I worded that with John. Anyway, let's parameterize this by mu2 and alpha. Neutrality and alpha deviation. We have epsilon being equal to 1. We have alpha being equal to 1 and mu2 being equal to 0, which means that all of our data has actually the same distribution. However, when we change those parameters, then we start getting some skew and we can notice that the distribution changes. Once we get to this point, from a scattered out. Not that similar from what we'd observed in our histogram. So what we really want, ideally, rather than revert to non-parametrics, to like within the neighborhood of the model. That's what we're aiming for. The most common way to In this case, for the location model, generalize this for the row function, row 1, taking the derivative of that, which is phi1, and then solve that bottom equation in order to find our estimate of mu hat. Now, you're already familiar with M estimates because, in fact, if we plot the psi functions, the psi function of the equation, then we get plots negative infinity, positive infinity, left to right, and the median, which is effectively a sine function. We can do the same for scale, albeit slightly differently computed. And in fact, we can have benefits because location is not scale invariant, is quite beneficial. So there are a lot of, and I have to kind of gloss over Huber was one of the first to propose a function parameterized by K. Tukey came along later and proposed his byweight parameterized by K. And the motivation that's often used in practice and used with median absolute deviation. Hampel came along later and proposed the location of contamination out on the tails. The three companies one of his many contributions to the field, the hyperbolic was green, but it's also optimal in another percent. And much more recently in this century, Bachmeier proposed location and scale estimate. These have two nice properties. First, there's no parameters, a lot less mucking about, but also it's a rejection point baked in 3.3. For a non-distributed outside of data. That's the idea. So there's a lot more. there's a large what we're showing. How do we know of your estimator? Efficiency is wrong. Already discussed the rejection point. What is interesting is that measures for the asymptotic value and variance are based on the true asymmetry distribution. What happens to these estimators when you have asymmetry? Not going to go into the definitions here, but obviously it will be a measure of the quality of the estimator, and less efficient estimators have higher variance, require more data for consistent results. Predominantly, again, I won't go into much detail, but proportional to the psi function. Psi functions without plotting the influence function. That gives us an idea of decimal of contamination at a particular on the x-axis. Importantly,
if we have a bounded psi function or influence function, we have a robust estimator. So we looked at the mean before, bounded, therefore it's not robust. Median is bounded, asymptotes, and therefore is robust. Derived from the influence function is the gross error sensitivity, which is an indication of the worst case of influence that contamination can give you. And complementary to that is the local shift sensitivity, which is a local worst case, how much it moves about in a particular location. Then when we look at the asymptotic variant, which was also proposed by Rosaire, tells us the influence of. However, in this case, negative, and it's positive values that we're more interested in. To give you some sort of intuition as to what these look like, this is a change of function, change of variance. A couple of key takeaways here is that we're just changing parameters. So we're moving the first parameter A out a little bit, and the result of that, we're changing B also. Wow, actually the last one's changing. Which is an important measure. I'm going to skip over that, called the change of variance. The worst case influence we have for the variance of our estimator. Hopefully you're still with me. We've talked about the rejection point. I'm not going to dwell on this too much. So what's the goal? I'm throwing out. We want to minimize the gross error, local shift, and change of variance sensitivity, and bring in the rejection point with sensible bounds, but we want to maximize efficiency. And there's a lot known about this already. The upper bounds for those sensitivities can be chosen somewhat arbitrarily. Maximum likelihood estimates are the most efficient, but they're not robust. The median is the most robust, biased robust, but it is inefficient. Three-part reader-sender, when we kind of optimal, and the hyperbolic tangent is optimal when we consider variance and the change of variance sensitivity. So that we can achieve really high efficiency here. In terms of optimality, we get comparable results. And a key thing here is that we're actually introducing a smooth. Which is what I want to talk to you about next. So this has been all preliminary. We're actually working on. So essentially what we've done is we've smoothed off this M estimator. Why do we want to do that? Because we get better results for the change of variance function and the change of variance sensitivity. We also end up with a Fréchet differential distribution. And we have definitions of the psi functions purely as polynomials, which makes them very easy to reason about and efficient to compute. The result, this is changed very, very simply, that we round off the sharp. This is the location, and the same true for scale. We're going to skip over these because it's just comparisons. And then skip to what I alluded to before, which is the measurements at the contaminated asymmetric distributions. This is just definition. Contaminated model. I'm not going to go into this. The goal here, what we're going to make our measurement Our hope is that our estimate and what we're going to do is we're going to keep the salad, so 5% contamination. Alpha, we're going to, and then we're going to change mu. And so what this looks like from a distribution perspective is just nudging the contamination over. Okay? So, result of that, just look at the plot on the this is the asymptotic value for location, tau 1 mu. We can see that this is a smooth three-part descender and the hyperbolic tangent optimal estimator give us the same equivalent asymptotic value. And it's essentially the same as we increase the spread or the distribution of our contamination. One important side note here is otherwise, the alpha. So we're actually just taking a more realistic variance. Now, Bartmeyer's estimates are lower, seem great. Why isn't that better? And the answer is because it pays a price. Smooth three-part redescender and the hyperbolic. It's a little bit. Again, we'd expect these plots as alpha goes to 0 to resemble the change of variance functions we saw previously. For scale, it's a similar story, value. And for variance, we see again, this is the as the spread of the contamination. Am I going for time? Sorry? Oh, okay. Wow. Okay, I'm flying through. So I'm sorry if I've gone too quickly. Okay, so how does it actually take that same approach to data where you nonlinear system of equations for mu hats or equation. We can use Newton's method, which is great. We can start from the median and median absolute deviation. However, we have a problem. We now have multiple roots because we no longer have convex functions. And this is a very real problem. And it's something that actually is statistics in general. There are a couple of solutions. One is or we can use a process called iterative reweighting. I'm not going to go into the it's an iterative process. Also, we start but we can fully iterate this procedure also with near zero risk of finding incorrect roots or not converging. For Newton's method, it doesn't converge often. You see the small samples, it won't converge. Reweighting is small samples, 5 onward. It requires more depth and is slower for large. So a work in progress to have a package where you just have a single function. And so to tie all this back together, to where we started, that green line is Here we show the and I think the key thing here is that it's just as simple once you have a function call to estimate a robust estimator for location. Okay, so in summary, we want to continue to use parametric statistics, but know that there can be contamination. There's no perfect estimator, but we can achieve high efficiency at robust estimates at the same time. And we've efficient, robust, comparable with our optimal by definition. Do have applications, but I'll skip over that and I will wrap up even a bit quicker. So if we skip back a little bit, then the asymptotic value for location indication. Even as the contamination moves into asymmetry, and that bias gets eliminated. Yeah, so you're right. The underlying um, robust. I have mental doesn't still do have a primary distribution that it distributes in this particular model. And if that is incorrect, but a model like web just may not act maybe. Yeah. Yes. Yes. Technically nothing. The cut reader sender is because it already has hyperbolic tangent estimator was to find but it is primarily based off of I'm not sure. We'll see Huber's psi function or Tukey in practice occasionally have. So by simply smoothing that function, which is already in use, we can get the benefit of close without. Yes. I have not. We have got a whole bunch of anyway applications that I listed a handful there, but we're actually working on robust adaptations for the lasso and applications of this procedure to that. So we'll do that first, but who knows, maybe we'll get to Bayesian process, what we can work on with that, all being well. Any other questions?
All right, well, that's the end. Thank you, everyone, for coming. And for those of you joining us for dinner, just follow the line so you know where to go.